Well, good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson, and I am the Executive Vice President here at Scripps Research. And let me be the first to welcome you to hear the lecture by Donna Blackmond, who is the chair of our chemistry department. Now, I did want to say a couple of things about Scripps and Scripps Research. There's a lot of Scripps institutions in the Mesa and in Southern California in general. We are all the benefactors, benefactees of Ellen Browning Scripps, who was a noted philanthropist. And the origins of the Scripps research come from the Scripps Metabolic Clinic that was founded almost 100 years ago. But she also founded the Scripps Institution for Biological Research and the Scripps Memorial Hospital, all roughly in the same time period. The Scripps Research, which is our Biomedical Research Institute, is the descendant of the Scripps Metabolic Clinic, whereas the Institution for Biological Research is now in the form of the Scripps Institute of Oceanography. And of course, Scripps Health is the descendant of the Scripps Memorial Hospital. So we all have common roots, but we are three very distinct institutions, and it's pretty understanding that some people might get the wires crossed. And I can't tell you how many times I've been, congratulated for having such a nice view of the La Jolla shores and the pier, and be able to go out on all the boats. I assure you that never happens. We are a basic biomedical research institute up here on the Mesa, just north of UCSD. So it's great to have the Scripps name, but we've all got to keep it all straight. So let me begin with just the briefest of introductions. Donna Blackmond is a noted chemist, and her specialty is analysis of chemical reaction mechanisms. So we're not going to talk about drug discovery today. We're going to talk about real basic research that has some pretty profound implications, for example, for the origin of life. And I'm going to let her elaborate. Donna has a couple of really special honors. So first of all, she was inducted into the National Academy of Engineering, but she was just about a month ago inducted into the National Academy of Sciences. Now, there's about 1,000 distinguished scientists in the National Academy of Science. There's very few who are actually members of two national academies. And there's a third national academy, which is Institute of Medicine. I'm not quite sure how I see Donna getting into that particular national academy, but it's a remarkable achievement to be a member of these two prestigious honorific organizations. Another honor another honor that she has is that she was just provided by Scripps an endowed chair in the name of John Martin. And unfortunately, John was a trustee and recently passed away, but we're really, really thrilled that Donna is the first holder of the John Martin chair. So Donna, I'm going to let you take it away, and why don't you grab the screen.
Oh, thank you very much, Jamie, for that very kind introduction, and welcome everybody to another one of our virtual front row lectures. Hopefully soon we'll be able to be seeing each other in person, but I'm very happy to be with you today to tell you a little bit about our research. Some very fundamental research, as Jamie mentioned, trying to understand the origins of life, how we all got here. But I also want to try to make the connection between that research and this search for better drugs that we're involved in here at Scripps, because they really are intertwined. So Jamie mentioned this word chirality in the rolling slides in the beginning, and I'll talk more about that. He gave you the definition, which I'll repeat, but this is the area of the facet of origin of life that I'm involved in, and it's also quite an important subject for drug discovery. But before we talk about that, we really want to talk about symmetry and asymmetry and mirror images. It's a field, or it's a concepts. They are concepts that fascinate not just scientists but lay people. If you've read Lewis Carroll's Through the Looking Glass, you remember that Alice was very fascinated by what was going on the other side of the mirror. And she wondered if she could get through to the other side, would her cat, Kitty, be able to drink the milk on the other side? Would it be too different? And she did go through and she saw lots of strange things happening there, opposites essentially. And we'll talk about that in terms of mirror images. So chirality, as Jamie mentioned, the word comes from the Greek word for hand. And it's called the property of handedness. And that's because if you put your hand up to a mirror, in the mirror, if you put up your right hand, you'll see your left hand in the mirror. So, every object has a mirror image. Well, okay, everybody except Dracula has a mirror image, but not all mirror images are chiral. In order to be chiral, you have to have an image that's asymmetric in the sense that you can't superimpose the object on itself. Like if you take your hands, your left and right hand, and you try to put them on top of each other, either this way or that way, you're always going to have your thumbs are going to be on opposite sides. So your hands are not superimposable. And we'll talk about what that means and what that means for drug molecules. But that concept isn't just for drug molecules, it's for macroscopic objects as well. For instance, screws, typically they're right-handed. Most screws are right-handed screws. And that is not superimposable in its mirror image. And you can't screw it into a left-hand socket for it. But or you could look at a nail, which is usually a straight nail, is superimposable. It's not chiral. So we call it achiral. So other objects around us, crystals, things like quartz crystals around the world. If you find a quartz crystal, they will look like this, and they are mirror images. You can actually see, we'll talk about this a little bit later, how crystals of opposite hands can be separated. In biology, there's a lot of examples of chirality on a macroscopic scale. You may have seen plant tendrils or wisteria climbing up a vine, or seashells. And typically in biology, one or the other hand tends to dominate. Whereas in abiology, like in these crystals, you tend to get an equal mixture of each hand. So this doesn't just happen on the macroscopic level. On the microscopic level, on the size of molecules, we have chirality. Molecules can have shapes that give them this property. What I'm showing here is alanine. It's an amino acid that's one of the 20 amino acids that you need in your body to make proteins and enzymes. And this is alanine. And alanine, looking through the mirror, sees its mirror image, but it can't be superimposed. The magenta amine group is going to end up on top of the blue carboxyl group. So that makes it a chiral molecule. And we call them left and right. We just do that. We name them. We have a naming rule. It's similar to how we named right-hand screws, or the right-hand rule in physics. The designation left or right isn't really have any significance in itself. It's just that we have to figure out, how to tell them apart. So we give them left and right names according to a rule. And these molecules are called isomers. If you remember from chemistry, isomers are molecules that have the same constituents, same structure, same chemical formula, but they have a different arrangement of the atoms. And these are a particular kind of isomer, which are called enantiomers, and that also comes from the Greek, the Greek word for opposite. So we talk about enantiomers, and we talk about enantio enrichment if we have more of one hand than the other. And so let's go back to hands again to sort of take this metaphor a little bit further. When we used to shake hands before the pandemic, I would put out my right hand and you put out your right hand and we shake hands. And that means hello, or it means yes, we have a deal. It's a message that we all understand. But if I would put out my left hand instead and you had your right hand, that would feel different. That would be a different message. And that's a very, very simplified picture of actually what happens in your body with a pharmaceutical or a drug molecule that's chiral can interact in your body differently if it's the left hand or the right hand. And the reason that it can do that is that inside your body, the receptors that are going to interact with the drug molecule are also chiral. And so they are going to see a left-hand molecule differently than they see a right-hand molecule, and they'll get a different message. And we'll see that can be quite important in some cases. For instance, with drugs, the left and right hand of a drug molecule, if it's chiral, they can act differently. One example is methamphetamine. Now we're going to go from the Greek to the Latin. We use the words levo and dextro, L and D for left and right, often with chiral molecules. But the left-hand molecule, methamphetamine, actually used to be in Vicks Vapo inhalers, and it was also an anti-Parkinson's drug. But the right hand of methamphetamine is what we all know of as crystal meth, which is not a nice drug. And if anybody here is a Breaking Bad fan, you might remember season 1, episode 2, before it went horribly wrong for Walter White. He was still teaching high school. And you can see he's using his hands. He's telling the students about, you can see on the blackboard, chiral, and actually behind it, non-superimposable mirror image. So he was describing the concept to his high school chemistry class. Because the left and right hand drugs can, versions of a drug can act differently, the consequences of that can range from being trivial to being tragic. And here's 2 examples. Ibuprofen, which we take for pain relief. You get the pain relief from the left-hand molecule, but both of them can cause stomach upset. So if we would make ibuprofen with just the left hand, you could take half as much. You get all the pain relief and half of the potential stomach upset. So that's not such a big deal as the second example here, which is thalidomide. And anybody who's my age or older will remember in the late 50s, early 60s, this was an experimental drug that was given to women, pregnant women, for morning sickness. The right hand acts as a sedative, but it turned out, they didn't know at the time, but the left hand drug caused birth defects. And so it was giving a very different message to receptors in the body. And essentially what the left hand version of that drug did was it caused capillaries that were beginning to grow in limbs to close up, to shut down. And so the babies were born no issues with the brain, but with deformed limbs. And so it was a terrible tragedy, which I will assure you can never happen again. This changed completely the way that we test drugs and regulate drugs. And so such a terrible mistake, tragedy could not happen again. But it just goes to show you how powerfully different the message can be from one hand of the exact same molecule to the other. And many pharmaceutical molecules are even more complicated, and they have many different places where they can be left and right. We call them chiral centers. This is a molecule that Merck developed as an anti-HIV protease inhibitor, which I worked on. I worked at Merck for a few years during the time they were being developed. It has 5 chiral centers. 5 places to choose left and right. That means there are 32 molecules that have that connectivity of the atoms, and only one of them is the active drug. So you can see that pharmaceutical scientists have to be very clever to make 1 out of 32. And the ways that they do that are some of the things that we're involved with also here in chemistry at Scripps. So chirality, it's not just drug molecules, but other simpler things like, well, before we go into other things, this is a list of some drugs that are chiral. Many of these, most of these are actually produced in an equal mixture of left and right. And some of them you may know: warfarin, a blood thinner. I mentioned ibuprofen, salbutamol, which is a asthma drug. Omeprazole, you can also get it as esomeprazole with just the left hand. But if we know that only one hand is doing the work, like we do with ibuprofen, why don't we just make the one that works? Why don't we just, stop making the other hand? And the reason is that that's actually really hard to do, because if you go into the lab, if I go into the lab and I want to make a molecule that's chiral, I will make an equal mixture of left and right. There's no difference there's no energy difference, there's no direction unless I give it a direction. We need to have some sort of a template or a pattern to drive, to push the molecule to go one way or the other. Think of it as like if you were going to make a pair of gloves. You need a pattern to make a left-hand glove, and that's different from making a right-hand glove. And so how do chemists do this? Well, the patterns that we use are called asymmetric synthesis or asymmetric catalysis. Asymmetric saying that we can actually make not an equal mixture, we can make just one hand, one side of the mirror for a drug. And what's very lucky for us at Scripps, we have at Scripps some of the best scientists in the world dealing with this property of chirality and for making organic molecules. First of all, Barry Sharpless, our colleague in chemistry, won the 2001 Nobel Prize in Chemistry for this very concept, asymmetric catalysis. He was a pioneer in showing how we can drive the reaction towards one hand of a molecule instead of making an equal mixture, to make either hand in some cases. And that's an extremely powerful tool that's used in pharmaceutical research today. I also have two other colleagues I want to mention, Phil Barrett and Jin Kuan Yu in my department, who've both been designated geniuses by the MacArthur Foundation with the MacArthur Fellowships. And they both work really heavily in this field of organic synthesis, trying to make asymmetric molecules. And most recently, you heard in the rolling slides, or you saw in the rolling slides in the beginning, the molecule ozanimod, which was developed by Ed Roberts and Hugh Rosen. And at first, it's now an FDA-approved drug for relapsing multiple sclerosis. But just, I think in the last month or 2 months, it just got a second approval for ulcerative colitis. And it's also still in trials for Crohn's disease. And that's the structure of the molecule. It has one chiral center, and it has to be enantiopure. So they have to direct it to make this hand and not the other hand. And we also have at Scripps Caliber. This is an institute that is involved in state-of-the-art drug discovery and development. And coupled with the science going on at Scripps Research proper, we're in the process of essentially making world-class drugs with this kind of research. And just one example, here's a candidate for prostate cancer treatment that they're working on, which is a chiral molecule. And obviously, this one does have to be enantiopure as well. So there's a lot of this going on at Scripps in various areas. And I want to tell you that not just drugs, but chirality can affect, is important in ways in your life that you'll know about. Chirality in the sense of smell. The left and right hand versions of a molecule can smell different. Now, actually, the molecule itself doesn't smell. The smell comes from the handshake between that molecule and the receptors in your nasal passages. And here's some examples. Carvone is a molecule that if you have the left hand, it smells like spearmint. If you have the right hand, it smells like rye bread. Limonene can be lemon or it can be orange. And citronellol, you probably know citronellol candles. That's the right hand of this molecule. And the left, if you have the pure left hand, it smells like geranium. It can also change the sense of taste. Again, it's not the molecule itself that tastes, but it's the handshake it makes when it meets its receptors on your tongue. And so here's a couple of examples. Aspartame is a sweetener, an artificial sweetener. It's the left hand that's sweet. The right hand has no taste at all. And it's the opposite for asparagine, which is one of your 20 proteinogenic amino acids. If you taste the left hand, it doesn't taste like anything, but the non-natural right hand is sweet. Glucose, the left and the right hand tastes just as sweet as the left. So it really is a very complex mixture of the molecule structure and the particular receptor, the chirality in the receptor that it's going to meet. And we can take that taste analogy one step further for any foodies in the crowd that have thought about or are interested in the concept of umami, which was really discovered in the beginning of the 20th century, but didn't really become a real thing that people believed in until much later. But now it's called the fifth taste. And it has to do with a synergistic action. First of all, glutamic acid or glutamate, you know it as MSG, monosodium glutamate, and it's a flavor enhancer. But if you put that together with the nucleotide of a G sugar like GMP, you get this really big umami effect. And essentially what happens in the receptor is when it binds the glutamate, you get some taste enhancement, but when they're together, it's really a powerful synergistic effect. And that's what gives you umami. And there's a few other sugars that do that. But it's a really interesting, to me at least, that it's L amino acids and D sugars that when you put them together, you get the amazing umami taste. And that actually fits with a lot of other things if you think about in biology, because all the building blocks for all living organisms are made up of L amino acids and D sugars. We're single-handed. Your body makes L amino acids. It doesn't make D amino acids. Your body makes D sugars. It doesn't make L sugars. And the proteins, these amino acids are the building blocks for proteins in your body, and they're all left-handed. And the sugars that go into DNA and RNA, and they're all right-handed. And the left and right doesn't matter, but the single-handedness, it never puts the wrong hand in a growing chain of amino acids or a growing polymer of RNA or DNA. And so that's a really important concept for life. And that really brings us to our research in this area. I've just told you that if I go into the laboratory, I'm going to make a 50-50 mixture. Biology knows how to do it where it makes only one or the other. But in the very beginning of the world, we call the pre-biology world prebiotic. In the prebiotic world, when the first molecules were being formed, how did they know to go left or go right? You know, we imagine that in what we would call the prebiotic soup, we would have an equal mixture of left and right-hand molecules. And so somehow we got from there to where we are today. And so the problem is really two parts. You have to break symmetry, and you have to somehow get away from that 50-50 mixture. And then once you've broken symmetry, you have to have a way to amplify that to much, much higher levels of enantiomer enrichment. The breaking symmetry part, we can pass off for now and just send the question to outer space. Chiral amino acids have been found on Earth that landed in meteorites. And they're always to the left hand. They have non-equal mixtures, and when they have an excess, it's always to the left hand. So you might say, so we could have been seeded from outer space, and that could have given us the symmetry breaking part. You could say that we're just passing off the question. We got to figure out where it came from out there. And there are a lot of people working, mainly physicists, who work on theories of like circularly polarized light in interstellar space. But here on Earth, you know, if we can assume we have a small imbalance, then we need to think about how we could propagate that. And actually, this idea of homochirality leads to the idea of biosignatures. A biosignature would be a substance or some phenomenon that would suggest evidence of life. And when the NASA missions to Mars, to other planets, and even in interstellar space, they are trying to search for, you know, is there life out there? We have to think, well, what would it even look like? And so one idea is there's several possible, you know, if you found a fossil, that would be great. But if you had chiral molecules that were enantiomer enriched, and if they were joined in chains of enantiopure molecules, like we have for our proteins and for our nucleotides, you know, that would be probably evidence of at least that life had been there. So that's one of the biosignatures that people at NASA are thinking of is the concept of chirality. So we've worked in this field actually more than a couple of decades, and we've looked at things all the way from purely chemical reactions that we could try to have this amplification of a small imbalance, all the way to pure physical methods. We have a number of different vignettes. I can't go through them all today. I'm just going to tell you about two of them. And we'll start with the physical side. And this is a vignette that I don't have time to go into all of the details, but it's something that you can ask me in the questions why I've named it chiral amnesia. But before we talk about that, we got to go back in time a little bit. We got to go back to Louis Pasteur. Louis Pasteur was a great scientist. You may have heard this quote that he has said, which is, chance favors only the prepared mind. And this man was a prepared mind. He was one of probably the most observant people ever to live. He's well known for some really amazing things. I mean, obviously we know pasteurization of milk. You know, before he developed the process to pasteurize milk, you know, I think up to 50% of childhood deaths came from diseases caused by drinking, you know, by having the bacteria in milk. And actually pertinent to today is that Louis Pasteur was also a pioneer in vaccinations. You know, he was one of the first to try to figure out how we could use viruses to kill, you know, to develop immunity against viruses. But what I'm going to talk about in his field was this concept of chirality, what he called the crystals of life. And he noticed in winemaking that wine, when you make wine, you can precipitate out. You've seen it in your wine glass, crystals, and they're tartrate salts. Okay, and so he went into the lab and he made some of these salts, and he crystallized them. And he actually looked at them and he realized that they had two forms that were mirror images. They're these hemihedral type crystals. So he took a pair of magnifying glasses and tweezers, and he separated them into two piles. And then he showed, I'm not going to go into everything he showed with how the two piles were different, but he was the first to realize that this property of chirality, he saw it on the crystal level, but he recognized that it was also on the molecular level. But then if you go back to his wine, what he saw with wine, the tartrate crystals that he saw in wine had only one of those crystals. And that's because that's from a biological process, fermentation. So he basically realized that this concept was really a distinct chemical difference between something that's living, that came from something living and something non-living. When I make something in the lab, it's not alive, so I get half and half of each. But if something comes from biology, it can be only one. Typically, it will be only one form of the chiral molecule or crystal. So we've done a lot of work in crystallization and various processes related. And so we devised a way that we thought we could separate chiral molecules. And so we called it Pasteur's tweezers revisited. It was a postdoc, Jason Hein, who's now a professor at University of British Columbia. And the idea was that we could take a pile of molecules, crystals, just like Pasteur's crystals. We took, it was one of the ones we did, and have an equal mixture of left and right crystals. And we mix it in water, make a slurry so that it's saturated, so that the liquid phase has the molecules in it too. Basically, molecules jump on and off the crystals into the liquid phase in a saturated solution. And we put glass beads in here and we ground this while we stirred. And so the crystals get smaller and smaller as we grind them. And then we put another vial where we had seed crystals of just one of the hands, just the blue ones are here, and they're bigger, okay? And we don't grind them, we just mildly stir that. Then we connected those, the two vials, and we let the liquid, we pumped the liquid back and forth, but we didn't let the solids move. Only the liquid could move back and forth, okay? And so then we did this, and after 16 hours, all the pink crystals are in one vial and all the blue crystals are in the other vial. And I don't have time to go into all of it. It may look like magic, but it's actually based on pure physical chemistry, really. It's the key is the difference in solubility between small and large crystals. Small crystals are much more soluble. You push a lot more of both crystals in this vial get pushed much more into the liquid phase. When they get over here, only the blue molecules in solution can crystallize out onto the blue crystals. So that's how we developed the separation process. The day we've done this for a number of molecules, but the day we decided to test this, Jason said to me, we got to do Pasteur's tartrate. That's the one we got to do. And so the day he set the experiment up, I came into my office and this was what was sitting on my desk. It's a pile of crystals, tweezers, and then the note says, Donna, you start on this and I'll race you. So we had a little bit of fun with that. And the concept behind this was written up in Nature News and Views. And it was a really nice thing to have somebody say that our method was the first original method for isolating single-handed crystals from a mixture since Pasteur used tweezers in 1848. So that was quite nice to hear. But actually, at the same time, we were thinking that separation is not as, if one of those molecules is the one we want to make the drug, we don't want to just separate them. It would be much better if we could just turn the wrong one into the right one, the wrong one into the correct one. And so we could use the same concept, but we just have to add one step. And that is we have to have a way of turning the molecules, the solution phase molecules, from left to right and right to left, an equilibrium process here. And that's well known to chemists. It's called racemization. And if we can add that to the concept that I just showed you, we can actually do this in one pot now. We put the two solids, equal amounts of each, put in the glass beads, grind, and watch this happen. With it, we put in a catalyst that helps us do this racemization, the interconversion process. And it depends on what molecule we do this with. It might be hours, it might be days that it takes, but we inevitably, inexorably get one enantiomorphic solid out of two. And here's just an example. We start with near racemic mixture, and over, in this case, over one day, say 20 some hours, we go all the way left or all the way right. And we've done it with a number of different molecules. The one I'm showing on the graph was aspartic acid. That's an amino acid, again, one of the 20 proteinogenic amino acids that you need in your body. But we've also done it with modern drug molecules. This is a precursor to Plavix. Plavix is $1 billion blockbuster drug for heart disease. And we can now, instead of having to throw away half of Plavix, if you make it in an equal mixture, we can turn one hand of Plavix into the other. And we actually collaborated with people in Holland at DSM Pharma, where pharma companies are starting to use this process We started out with this idea about origin of life. How did we get here from the prebiotic soup? But it also takes us into processes that are actually quite valuable for modern pharmaceuticals. So the next vignette will be more about the chemical side of things. And this is what I call the chicken or egg question, which I'll explain by the end. And it goes back to the smallest molecule that can be chiral. I call this the primordial chiral center. For the sugars, it's glyceraldehyde. Glyceraldehyde is a 3-carbon sugar, has 1 chiral center. If you can fix that to the D sugar, then you're on your way to making D-ribose, which is what our life form is based on, and deoxyribose. For the amino acids, the first chiral center can usually be traced back to an amino nitrile, as a molecule that we can turn any amino nitrile into an amino acid. And that's been known since the 1850s. It's called the Strecker synthesis. So, but the point is that we know the sugars and amino acids are the basic building blocks of biological homochirality. And our work is now showing that it's synergistic. The sugars help the amino acids help the sugars. And I'll show you that in just 2 very, very brief vignettes. This is a case where the amino acid mediates the sugar synthesis. This reaction is a reaction that was developed a little more than a decade ago by John Sutherland, and at the time his student Matt Powner, to synthesize RNA. This is a precursor to make an RNA monomer, and it's very prebiotically relevant. One of the big problems in origin of life chemistry is trying to understand how we get to RNA. We can find ways to make ribose, we can find ways to make nucleotides, but we trying to put them together has always been a challenge in any reaction that would be of relevance to what could be done in the prebiotic world. And so this made a big splash, this paper that John Sutherland published. It was highlighted in the New York Times science section, and it was a real breakthrough for people working in prebiotic chemistry. But what they did was they started from D-glyceraldehyde. They basically didn't try to address the chirality question. They just started from the D. And so our contribution to this field is to try to understand if we could still get there, but do it with an amplification mechanism. So we could start with an equal mixture of left and right glyceraldehyde. And that's what we did. By putting in an amino acid, we screened a whole bunch of amino acids as mediators of this reaction. It turns out proline is the amino acid that does the best work for us here. And we could watch over time, we start out with a 0 excess. Basically 0 means 50-50. There's no difference between the left and right. And we get to a very high excess of D-glyceraldehyde over time, over a couple of hours, really. And then, but at the same time, when we were doing this in the presence of the other molecule, the 2-amino-oxazole, and that gives us this RNA precursor at very high EEs, and we can crystallize that. Jason Hein, the same postdoc, made this beautiful crystal of this molecule. It's called riboamino-oxazoline, and it's 2 steps away from RNA, and this is the structure of it. And so we could start with a 50-50 mixture of glyceraldehyde. And all we needed was a small amount of an L-amino acid to get us to RNA. And this was actually really important work because it gave, especially their synthesis, even before the chirality piece, it gives support for a hypothesis which is called the RNA world hypothesis. And the idea is that we know our life form is based on DNA. That's where, that's our main genetic polymer. But it turns out that RNA is a pretty cool molecule itself. We know that now with vaccines, but it also, it could have been a life form because it has pretty much all the characteristics that we would say we need for life. It can self-replicate, it can make itself. It can do things. It can catalyze other reactions, and it can and it can mutate. It can actually undergo Darwinian evolution. So those 3 properties say that maybe very primitive life forms were based on RNA rather than DNA. And so John Sutherland's work showing that you could actually make RNA in a way that the prebiotic world wouldn't have been outlandish was really support for that hypothesis. So this is an amino acid mediating sugar synthesis. So now here's a sugar mediating amino acid synthesis. We start again from the amino nitrile, as I mentioned, is the first chiral molecule on its way to amino acid. And this is alanine. It's the same, it's a precursor for alanine, the molecule I showed you in the mirror in the very beginning. And we add ribose, D-ribose, as as a catalyst, actually, and we make enriched L-amino acid in very high enantioenrichment over time. And we go through, you can see we get up to quite high levels. We go through this intermediate, which is an amino sugar. And we have completely characterized it by experimental techniques and by computational work. And it's really interesting because it's reminiscent of a lot of there are amino sugar molecules like in the membranes in many in your body, and essentially so the same things that we think about for the origin of life actually also are carried over into life forms today. And so the bottom line really here is that this emergence of homochirality and sugars and amino acids, they're intimately connected to each other. And so that brings us back to, chicken or egg. I can't tell you whether the sugars came first or the amino acids came first, because we know that D sugars can enrich L amino acids. L amino acids can enrich D sugars. So you can take your pick. I think they were synergistic and probably iterative to get us increasing, levels of enantioenrichment until homochirality emerged. So that's actually the end of my scientific talk. I'll leave you with that as you can ponder the chicken and egg question. And I'll just finish by, I tried to name some names along the way, but there's been a lot of people involved in this work since I was working on this before I came to Scripps. 11 years ago, I was at Imperial College London doing some of this work. And so there's been funding both from the other side of the pond and here. And again, I want to especially mention the John C. Martin Endowed Chair, which I was just awarded. And actually, the last thing I want to do is just take a minute to mention John Martin, who was on our board of trustees, amazing scientist, a giant, it says here, a giant of our space. He was the CEO and president of Gilead until he retired. He is responsible for saving millions of lives. His work on HIV turned into the cocktail drugs that turned HIV from being a death sentence into a manageable disease. And he also cured hepatitis C. So, he was innovative and creative. And actually, he was an engineer and a chemist. So that speaks to my heart. And I think we want to take a minute to remember him and what a great friend he was. He and his wife, Lily Lou, in supporting Scripps. And so we're grateful for both of them and for all of our donors. So I'll just end and just mention the word chirality again, since it's really, my thing. And we'll be happy to take questions, but I'll just put up before we go to the questions to give you the highlight for what's coming up, which is our next seminar in the Front Row series will be in August. That's Howard Hein, one of our newest professors, and it says Department of Immunology, but we're going to claim him for chemistry as well because he has a joint appointment in chemistry, and so this will be a really interesting seminar about the microbiome and how do we boost immunity, not just against infection but also cancer.
Terrific. Thanks a lot. So we have quite a few questions to go through. I think it stimulated a lot of interest. So one of the first questions that came up is from Hui Pham, and he's asking, are there major sources of chiral molecules besides drugs? You kind of started out talking about drugs.
Yeah.
But what about other natural products that come from fungi and bacteria or toxins?
Yeah, absolutely. I mean, natural products are, we call this the chiral pool. I mean, how do we get, for instance, Barry Sharpless's work on asymmetric catalysts to make more chiral molecules, he had to have some chirality to start with. And so we have what's called the chiral pool, all the natural molecules out there. And some of them are incredibly complex. Some of them are simple. Something like alpha-pinene is a chiral molecule found in nature, which finds its way into a lot of the reagents that people use for making more chiral molecules. Yeah, so the world is very rich, and people go on expeditions to find natural products in the ocean and in the tropics, and it's incredibly rich world. And then we have scientists at Scripps who are involved in natural product synthesis, trying to find ways to go in the lab and make these molecules that we find in nature. And we have,
it's safe to say that in biology, chirality is the rule rather than the exception. Yeah,
there are some interesting exceptions, and there are some post-translational changes in amino acids that change them from L, the normal one, to D. And there are if you look at archaea and bacteria, they split somewhere early on, and archaea actually have some cell membranes that are all D amino acids and not L. So, you know, that's actually a really intriguing question for thinking about, you know, when the decision was made, how far back and where it split. But yeah, there's chiral. Your world is chiral. Your world is enantio enriched.
So here's a question I'll paraphrase from Mahak Bhushan. Basically, you were talking about the origin of life, how do we get the enantio enrichment? And is there an underlying cause, or is it just like by chance the first one to work happened to be
right?
left or right?
And that's a huge debate. If you go to these meetings, because nobody has the answer and no one has a time machine, you can get some really vociferous debates going because nobody can prove the other guy wrong at meetings. But so there's a group of people that think, some people think it was just chance, that maybe there were chiral pools starting up, say some enantio enrichment in different places, and then the big one ate the small one. And some people think, no, we were absolutely predestined to be L amino acids. And they look at things like some things in physics, like Coriolis forces or parity violation energy difference. None of these things have ever been proven, you know, to be the cause. But and if I had to say, I think that I would say it's a toss up that we became L amino acids versus D sugars versus D amino acids. But once we are L amino acids, we're definitely D sugars. Or if we were D amino acids, we'd be L sugars. It's my feeling from the work that I do. But it's a question that, you know, it's great for, you know, going to the pub at the end of the day of meetings, but it's not one that I know, it's not one that I know of a way we can solve in the lab.
Yep. Okay, here's a question from Rob Statler. So you talked about how there was an initial choice of chirality, but then when you get to something like RNA, there's actually 4 adjacent chiral centers in the sugar. And so there could be up to 16 different isomers in this. So how did that one get picked after? So
that's it. Yeah, if you go to try to make ribose, if you have the glyceraldehyde, when you say D sugar, you mean that it's the chiral carbon that's going to be attached to the phosphorus. You know, it's going to be on the end of the chain, and the other sugars, the other chiral centers, can be D or L, and they would be called diastereomers. And of the pentose sugars, which is what ribose is, there's ribose is what we have in our life form, but there's arabinose, xylose, and lixose. And so we've been looking at ways to make, you know, and just the thermodynamic you know, mix of those would give us mostly arabinose and not very much ribose at all. So it's still an open question how we got to ribose. It's not the same issue, because when you talk about making enantiomers, you definitely need a pattern, because they're exactly identical, like your left and right hand. If you have more than one chiral center, they're not exactly the same. They can have different boiling points or melting points. So you can use things like crystallization, which is what we used when I showed you the riboaminoxazoline. It turns out that the ribo form that was made in the Powner-Sutherland synthesis crashes out, is much less soluble and crashes out before anybody else. And so crystallization helped chemistry win, you know. And so I think that a lot of people now are thinking that we can't just think about a chemical reaction. We have to think about all the physical processes that and that may be one reason that we got here is that molecule crashed out, was left behind when the water washed over the pond, and then that one got dissolved in the next rainstorm and it became life.
Mm-hmm. So you mentioned NASA, and one of the things, there was the very first Mars mission some time ago.
Yeah.
They landed a rover, and one of the experiments they did was they sort of scooped some soil back into the rover, and then they added sugar and some nutrients, and then waited to see if anything would grow.
Yeah.
And I remember thinking, what if you got the hand wrong? So they put in D-glucose.
Right.
And what if Martians eat L-glucose? You know,
right.
So is there any kind of insight into this?
Yeah. Well, so that experiment, those early missions, they went out thinking, you know, we're going to find life, and then that didn't work. And they came back, and it was actually a few years before they really got back on their feet because everybody was so disappointed. They realized they didn't even know what question to ask. Right. What is it going to look like? And so that became this whole area of developing what would be a biosignature. Okay, and so doing experiments out there is pretty challenging. So now the Martian mission that's out there now, Perseverance, it's going around and it's collecting samples in these, they look like thermos bottles, and they're going to leave them there. Okay, they're making a pile of them. And then in about 10 years, another mission, because it's not coming back, that mission, but the next mission is being designed to come and pick those vials up and bring them home. And then we have the material to do experiments on, kind of like we got from the moon. But that'll be a long way away. But yeah, it is a even deciding what would it mean. I mean, there's been a couple of papers recently out talking about, there's one person has a theory that if a molecule is over a certain molecular weight, it's life. Well, then my Coke bottle is life. My plastic Coke bottle is alive. So even people even deciding what it means to be alive is not a completely settled question.
So there's a couple of questions. I'm going to kind of aggregate some thinking, but so the question, are RNA and DNA chiral, and what's their relationship to protein chirality, and how does the protein enforce the chirality of the nucleic acids? So, and then maybe if I would just ask you to comment, there's this whole kind of universe of people making mirror image proteins.
Yeah.
And maybe you comment on that as part of your response.
Right. So what I was trying to show about the work that we're doing is that I think that once you are L-amino acids, you are going to be D sugars. I mean, that's what our work is seeming to show. So the DNA and RNA are made up of D ribose and deoxyribose. So they're the D sugar, and we have the L-amino acids. So I think that basically, as life started, as we started to get the building blocks enantioenriched in this way, then that's when the molecules started learning how to shake hands. Basically the handshakes that's the message that tells the molecule what to do, was developed from those building blocks that were major, that were dominant at the time. And that just kept evolving into our amazingly precision life form now with, if any of the talks that we heard in this series where you talk about this receptor and that, and all the stuff that goes on, it's hugely complex, but it had to start somewhere. And I think as soon as we started to get enantioenrichment in our L sugars, and at the same time, L amino acids, at the same time, D sugars, that was where it was set what our handshakes are going to be like for the rest of our life form. And again, people are making the opposite hand for a lot of reasons, to make sensors, so if you make a D nucleotide, you can use it for things that your body won't chew it up. It won't recognize it. It won't try to say, wait, you're the wrong guy. It won't try to do the same thing with it. So there's a lot of stuff about like antisense. There's a lot of reasons to try to make the opposite hand. Mm-hmm. Now there's one person, George Church at Harvard, who's actually trying to make an entire world on the other side of the mirror. And he's making molecule after molecule, basically trying to make a whole mirror image world. I don't know how that's going right now, but
I'm sure it's a challenge. The hard part will be making ribosomes.
Yes, there you go. Well, if you got them all, if you got all the components, I don't know.
So here's a question, and I think it's kind of intriguing and topical. Is chirality involved in the mRNA vaccine?
Well, the RNA is a D sugar. It's basically, a ribonucleotide, like I showed. It's actually, that umami molecule, the GMP, is, that's an RNA monomer, right? And right. So, basically, the molecules I showed are building blocks for RNA, and it's what happens when it gets to be mRNA or another kind of RNA. It's when you build up enough of these molecules with different bases, same sugar, but different bases. Then it has a chance to be informational. It says, if I have these bases in a row, it means do this. If I have those bases in a row, do that. It's a code. Mm-hmm. And basically you have to that's why you need a polymer. If we found an enantioenriched amino acid in outer space, that would not be enough for me to say there was life out there. That if I found an enantiomer-enriched, RNA polymer, I would say, yeah, that's kind of hard to do without life. And so, all the sugars in the RNA are right-handed, and the differentiation in terms of what type of RNA you make comes in the bases and how they're, in the messages that the different bases in a row are telling.
Here's a question from Peter Tobias. Hi, Peter. So he asked the question, why are large crystals less soluble than small crystals?
Okay, so yeah, it's the Gibbs-Thompson rule. Okay, when we talk about solubility, when you learn it in physical chemistry, you're talking about a completely planar surface, a flat, infinite surface. That's the solubility. But if you have a small ball, you can think of it also in terms of phase changes, like a drop of water on a window. A small drop will evaporate faster than a big drop, right? And so a small crystal, it has to do with surface tension. A small crystal.
Isn't it surface to volume?
Surface to volume ratio, right? You have a much higher surface tension, so the molecules in small crystals just don't want to be there. They want to jump off. Whereas if you have a big enough crystal, if you had a big enough crystal that it was a complete flat plane, then you'd be back to the total the theoretical solubility. And so smaller crystals dissolve. There's kinetics and thermodynamics. Smaller crystals dissolve faster, and bigger crystals want to grow more. So the molecules from the small crystals went into solution, and then they saw a big crystal, and they said, "Oh, we got to go down on that crystal because there's too many of us in solution." So that's essentially how that process almost becomes autocatalytic.
So you mentioned in your introduction, or no, it's in your seminar, you mentioned the contributions of Barry Sharpless.
Yes.
So Barry really developed the first way to turn a symmetric molecule into an asymmetric.
Right.
And he was duly awarded the Nobel Prize. So maybe in closing, I could ask, what are the synthetic chemistry challenges? What remains to be done in terms of introducing chiral functionality that would be Nobel worthy?
Yes, well, I will tell you that if you'd said this 10 years ago, we would have said CH functionalization will never be able to do that. Taking a simple inert carbon-hydrogen bond, and that's not different from the ones around it very much, and making it differentiated. And I can tell you today that I'm fully of the belief that our colleague Jin Kuan Yu will win the Nobel Prize for his work. He has just done tremendous work over the last decade in showing how we can make asymmetric molecules out of very inert, very non-distinguished CH bonds. So CH functionalization and Jin Kuan Yu and his contributions. That's the answer.
I think we should have a front row for Jin Kuan.
I think we should. Yeah.
Incredibly brilliant, energetic speaker, and I hope you're right about his future prospects.
Yeah.
Okay, thank you, everybody. I think we're going to draw it to a close. It was really terrific to hear about your work, Donna, and its wide-ranging implications from the drug discovery through the origins of life. I ask you all to tune in for the next front row, which is August 18th. Again, it's Howard Hein, and he'll be talking about the interaction of the microbiome with your immune system. So again, thank you all for coming to the front row. I hope you enjoyed the presentation. Spread the word, and see you next time.